Morning, everybody. Welcome to Grand Rounds. Just two announcements I wanted to make. Is one which was that last Friday was match day when we got the results of the match, and as usual, we had. I mean, it's a very happy event because we always sort of have tremendous trainees coming in, and it's very exciting and invigorating. And this year was even more so. We really did extremely well. Not that we didn't expect to, but it was a transition year. So one could say that those kinds of things could have unexpected or unjustified effects, but in this case, it certainly didn't, or if anything, was the opposite. So congratulations to the training team and everything, and this now follows on the child match, which occurred, is Oliver here?
Yep, January 4th.
Yeah, and that was, comparably so. The country may go to hell, but we're bringing good people into medicine. The other thing is, just wanted to, we don't usually mention things like this, but Bud Rowland, a former chair of neurology here, died this past week. And Bud was an extraordinary individual, as a great academic neurologist and chair at Columbia. But I knew Bud in a variety of ways over the years, and apart from being an extraordinarily gracious person, in terms of the interaction between psychiatry and neurology, which is a field where we're kind of estranged siblings, it was he was dealing with him was like you were part of the same discipline. There was never any real sort of distinctions or anything, and if there ever was to be any kind of greater integration, Bud was certainly the kind of person who would have been very effective in terms of facilitating or welcoming that. So today our grand rounds is from one of our own faculty, Jeremy Veenstra-VanderWeele, and I think when Jonathan Posner suggested Jeremy, I said, he hasn't given grand rounds already? And how long have you been here? 2+ years. So in any event, we thought definitely needed to schedule him. Jeremy hails from Canada, Ontario, and then went to college at Harvard, and then medical school at the University of Chicago. And following that, he did his training at the University of Chicago, and also the University of Illinois, and then was recruited to Vanderbilt by Stephen Heckers, where he was. Were you recruited as the division director, or you became the division director?
I was a postdoc.
So he came there as a postdoc. He was on the faculty, and then by somehow drafted in or appointed or selected to be division director. It seems to be your fate. You go places and to pursue your research, and then you, no hints there. And at Vanderbilt, working very nicely with Stephen Heckers and in the department that was developing, we spotted him, or more specifically, Maura spotted him and said, this is somebody we want, and went out and recruited Jeremy. And he and his family moved east. They're Midwesterners originally, but I think have adapted comfortably. And made has made an important contribution to the department. Jeremy is an MD, PhD, and has had a focus in what might be sort of developmental neuroscience and genetics of developmental disorders, with a particular focus on autism. And in addition to have been a highly attractive academic psychiatrist to recruit, I think he also is really exemplary of a process, which is that when I was training back in the sort of Middle Ages, there were only very few people who did stuff like Myron Hofer did, which was to be a psychiatrist, but have a background in some laboratory-based life science discipline. And in Child, there were none. And Child has sort of followed as general psychiatry has progressed to the point that it's now being populated with more such individuals. The kind of early or kind of standouts in that way were people like John Rubinstein. Is Dominic Ciarello a child psychiatrist? Does anybody know that? Huh? Roland.
Yeah, he was a child psychiatrist.
Yeah, so this is a Stanford child psychiatrist who was appointed NIMH director and then unfortunately died. But then, you have Matt State, Alan Rees. So there have been individuals, the maturation of the child discipline, is progressing, and I think, Jeremy is clearly an exemplar of it. In coming here, he plays an important role because he's really has positions in multiple critical programs. He's a member of the Sackler Institute, and he's also integrally involved in the Center for Autism and Brain Development, which is the program that we have with the hospital jointly with Cornell and is based on the Westchester campus. So it's great to have him give grand rounds now, and he'll be speaking on what he's described in his title. So please welcome Dr. Jeremy Veenstra-VanderWeele.
Thank you, Jeff, for the very generous introduction. I have to say, among that generosity, one thing, I actually have PhD envy. I didn't get a PhD, so we can talk about that in the psychodynamic portion of the question and answer session. This is sort of a positive and negative title in one, and I'm going to talk about some sort of work in preparation. I'm sort of pinch hitting, learned I'd be filling in about a month ago and thought, what can we talk about here? Imagined talking about some of these stories that are in development maybe a year from now, but I'll tell you where we are at the moment. Before I do that, I think it's important to note some disclosures. I'm going to mention some work that was funded by the pharmaceutical industry. None of that has come to market, but I think it's relevant as you're evaluating what I'm saying. And then because I'm giving this as a grand rounds and thinking fairly broadly, I'm not going to call out a lot of individuals. And so I was horrified to find that I have a very out-of-date picture of the lab here. So apologies to folks in the lab who are not on the picture. We have another one scheduled next week. So I'm going to very briefly describe one of the core challenges in autism spectrum disorder before really pivoting to talking about treatment. I'm only going to talk about current treatment really to frame some of the challenges as we think moving forward about how we develop new treatments. So really I'm going to be highlighting the negative side of that. It isn't that we don't have effective treatments, but I think it's important to think about those challenges. And then talk about how we move forward. And this includes what is usually described as precision medicine, which is taking mostly rare genetic findings and carrying those forward to have an understanding of neurobiology that leads to molecular targets for treatment. And then talk about almost the opposite approach, which is to start with symptomatic targets and to move underlying those symptomatic targets toward approaches that may apply across species perhaps, but don't necessarily relate to the causes of autism spectrum disorder. And then talk about maybe how we can work between these two approaches using biomarkers that are more common than the extremely rare genetic variants to tie groups together who may benefit from common treatments. I'm barely going to mention behavioral treatments, okay, which is kind of embarrassing because that's where most kids with autism spectrum disorder should be spending most of their time. Those are the treatments we currently know work, but this talk is really focused on biology, and those treatments aren't necessarily based on an understanding of neurobiology. In the end, I'll come back to that and talk about how we're going to need to put things together, which is an additional challenge. So I think it's fair to start with what is autism spectrum disorder? So it isn't really an entity, right? This is something that is a description of common symptoms. You can think of these as sort of a common final pathway that can arise from many different underlying risk factors. So when I see a child with autism, I'm usually thinking about that child from this perspective, where you see autism symptoms as diagnosed by the DSM-5 in the center, and it's critical that the child meets those criteria, but I never see a child who only meets those criteria. Usually they have some degree of cognitive disability, often behavioral comorbidity, and that's usually, as a child psychiatrist, what I'm pushing on. Medical comorbidities are common. Increasingly, kids are walking in with genetic diagnoses or walking out with genetic diagnoses. Now, those are well-defined entities as opposed to things that we are defining based on very careful behavioral observation, but still behavioral observation. And then a number of biomarkers that as yet haven't really translated into the clinic. So when I tell you about a treatment study in quote unquote autism spectrum disorder, that treatment study may include a kid who fits this pattern, a kid who fits this pattern, and 148 other kids. And so we have to be very clear about what we're actually doing in these studies, and we don't have people who necessarily have a common etiology. I'm going to use this pattern and this pattern as examples through the course of the talk. So when we think about autism risk, most of the epidemiologic data still favors a model where you have multiple common risk factors. These can be genetic or environmental that coincide in an individual to lead to sufficient risk to confer diagnosis. In truth, however, what mostly has been identified are really quite rare events, mostly genetic, that confer the majority of an individual's risk. None of them are strictly deterministic, but if you took these away, the child would almost certainly no longer have autism spectrum disorder. So the most common of those that I'll use as an example today is Fragile X syndrome. About 1% of kids with autism spectrum disorder have Fragile X syndrome that we'll talk about in more detail. More of these sorts of things are emerging over time as genetic studies are pulling apart the syndrome. So people are starting to identify variants in kids who don't have syndromal features. And one of the early examples of this was chromosome 16p11 deletion syndrome. That's about 1% of the overall population. As these accrue, none of them really exceed that 1% level, which is a real challenge when we think about trying to pull treatments together. What we hope for is that we're able to also identify environmental factors that perhaps coincide with this. And if we can pull those together, maybe we'll have clusters that will tell us about a common pathway. There are emerging examples of this with mTOR signaling that maybe then can be turned back to the general population of kids with ASD who do not have one of these really quite rare variants. So that's the simplest piece of things, unfortunately. You know that genes, by and large, come first. They're there from the very beginning. As we move forward from tens of thousands of genes, with evidence accumulating for a couple hundred implicated in autism spectrum disorder, you move orders of magnitude up to synapses, quadrillions, and then behavior is essentially infinitely complex. And then you have to think from a developmental perspective. So that obviously gets a lot more complicated than simply identifying risk genes. Now, as we move from an understanding of risk genes to how that risk is realized in cognition and behavior, we've selected at the level of cognition and behavior, so you should see convergence again. And I think that that is the hope moving forward. We're not there yet in terms of understanding convergence across models of autism spectrum disorder, but that's the idea, that perhaps you have convergence up here that expands to a treatment that helps a broader swath of individuals. Instead, most of the convergence that's been identified to date is really at the level of protein networks, with a black box between that and cognition and behavior. Now, that's a big improvement on our previous pathway to treatment in autism spectrum disorder, where we just had a big black box. And we should be honest, all of our treatments in psychiatry really arose from a black box model of neurobiology. That isn't to say we don't understand something now, but certainly when the treatments were initially identified, it wasn't based on targeted treatment. An additional complexity, and Huda Zogby really articulated this well when she visited, is that almost without variance, if you identify a genomic region that is deleted or disrupted in autism spectrum disorder, if you look long enough and hard enough, you'll find other individuals in which that same genomic region is duplicated or where you see gain-of-function mutations. And that's really troubling. Not all cases see autism on both sides, but certainly developmental difficulties on both sides. And if you think about treatment, which is where I'm oriented, if you improve things for this individual, you may actually make things considerably worse for that individual. So that's a real tension as we think about moving forward with treatment development. So I'm going to give a fairly brief overview about what we know now about autism treatment and how we know it. So behavioral interventions, as I mentioned, this is where most kids should be spending most of their time. I have just a couple slides on this. So we know that early intensive behavioral intervention is effective for many children with autism spectrum disorder. There are also some targeted behavioral interventions that I'm really not going to cover in any level of detail. This is really the best study of an early intensive behavioral intervention. You can see it's a pretty small sample size. And what improved over the course of 2 years of intensive treatment was IQ score, so that you see that the treated group moves up versus the group that was treated in the community as a comparison. It's a meaningful difference. So we're talking about half a standard deviation of IQ, maybe a little bit more. But then if you look over at other things like adaptive behavior, you can see that the treatment as usual group really saw a decline versus peers, whereas the treated group showed a maintenance of their current level of adaptive behavior compared to peers. Again, that's a real positive, but these are not core symptoms of autism spectrum disorder, and that I think is an important lesson. It may be easier to pick things up with direct observation, with direct testing, than it is with ratings based on behavior. On the medication front, we have good evidence that atypical antipsychotic medications, as they've been described, though they clearly do more than just serve as antipsychotics, are calming in this population. They help with irritability and agitation symptoms. They also obviously are difficult to use. In individuals with irritability and agitation, you also do see improvement in some other symptoms, but it's really difficult to pull apart whether that is specific improvement or whether it just means they're no longer so agitated and can do better in other domains. So this is one of the larger studies of aripiprazole, where you see the placebo group shown here, ratings of irritability and agitation here on this axis, and you see 3 different doses over time. Clearly there's an advantage versus placebo, but it isn't a huge advantage, and actually it's a pretty substantial placebo effect in this study, even though you're looking at quite severe symptoms, not symptoms of autism, but irritability, agitation, kids who are hitting, self-injuring, having tantrums. I'm just going to briefly mention that abundant data now shows that it's possible to successfully treat ADHD in the context of autism spectrum disorder. I'd be remiss not to mention it. It's sort of the rule of 50%, where half of kids get better on each one of the 3 options that are usually turned to, and more side effects are generally seen than in the general ADHD population. And then this has been a puzzle. So repetitive behavior is one of the core symptoms of autism spectrum disorder. And we know something about treating repetitive behavior, at least in the context of OCD. Early reports showed that fluoxetine and fluvoxamine, which is actually the one studied first, helped with compulsive-like behaviors in adults with autism spectrum disorder. But in children, where the repetitive behaviors were lumped together, and by and large, the children were unable to describe the behaviors in a way that was consistent with OCD, there really hasn't been much support for this. And this is another place where you can see the challenge of the placebo effect. So this is the largest study done. It was a study of citalopram by Brian King and colleagues, where kids took either citalopram or placebo, and the ratings shown here are clinical global impression of improvement. So physicians simply saying, is the child much improved or very much improved or minimally improved or not improved at all. Initially, they saw no difference between the groups, and they went back because a number of us said, well, clinically, we don't just lump all repetitive behavior together. Generally speaking, we only treat repetitive behavior when it's distressing or interfering in a substantial way. So they went back and they did a median split analysis on 3 different domains. So one of those domains was disruptive behavior, another was caregiver strain, another one is a little harder to parse, which was general mood and autism symptoms. And in each case, when they looked at the kids who were less affected, the bottom half, they saw a differential response to placebo versus kids who were more affected. Sort of discouraging when you go back and realize that you've distorted your study by including some of these milder kids. And if you look in the more affected kids, there might even be a benefit for active treatment versus placebo, giving us, unfortunately, another lesson that we probably need to enroll kids who are more affected in whatever domain it is that we're interested in. So, and then this is the evidence that's sort of pro-treatment, but there's also abundant evidence for another medicine that has actually more randomized controlled studies than any other drug in autism spectrum disorder. Anyone know what it is? Who hasn't heard my talk 3 times in the last month, like Bill Pfeiffer? It's a different talk each time, but still. Any guesses? So risperidone, that's what most people guess, and it actually isn't risperidone, although there is abundant evidence that risperidone is helpful. It's actually secretin, which we now know with tremendous confidence does absolutely nothing useful in autism spectrum disorder. You have a patent on secretin. So we'll have to talk later. This was probably taken by tens of thousands of kids in this country after a published case series and a lot of media attention. There were then 7 reasonably well done randomized controlled studies. And what we really learned in those studies was that the placebo effect is remarkably powerful. In some of these studies, placebo effect was up to 50%, where kids were clinically significantly improved. And this was one of the better studies from Jim Bodfish and colleagues, where they looked at the secretin group shown here and the placebo group shown here, where clearly placebo is, if anything, outperforming, and they looked at any number of scales. This is sort of the most comprehensive scale of autism-related behaviors. So clearly no benefit for active drug over placebo. But this is really a searing quote from that paper in the New England Journal of Medicine. Even after hearing these results, nearly 70% of parents were still interested in this as a potential treatment. So, that makes us all feel like, why are they so irrational? We don't really have much to offer. And so when you see results of this study, and you're desperate, you think, well, maybe if it was done a little differently, maybe if this was done, maybe if more doses were given, maybe it's the wrong dose. But this is really a challenge for us, that we have to base our conclusions on very careful placebo-controlled studies because we know the placebo effect has such a big impact. So this is, again, from Brian King and colleagues looking at placebo response across many studies in autism spectrum disorder. You see as high as 50%, and you see really there's a clustering around a 30% placebo response, which is really confusing. 2 decades ago, you could publish an open-label medication study in autism spectrum disorder. In the Orange Journal, there were even a couple of them published in the American Journal of Psychiatry because no one thought autism improved. Right? So of course you don't need a placebo group. It may even be unethical. But we understand in other disorders that placebos have a substantial impact. And this can move any number of things, including things that we usually think of as purely medical, things that you wouldn't imagine would improve based on hope alone. And we know that some things increase response to placebo. So intensity of treatment like secretin injections, cost of treatment, probability of active drugs. So if you're more likely to be taking an active drug, you actually, if you get slipped into the placebo effect, have a bigger response. That may be part of the answer with that aripiprazole study that had a big placebo effect. Subjective outcome measures, and then inverse illness severity. So the less ill you are, the bigger the placebo effect is. And that's really been a challenge in the depression literature. So it isn't just that, however. And in ASD in particular, I think it's important to realize that the placebo isn't just the effect of giving the placebo. It also is a way to measure nonspecific change during treatment. So we all fluctuate, right? And who enrolls in a clinical trial when they're at their best? We more or less enroll in clinical trials when we're at our worst. And then you expect to see some level of improvement. It also is the case that just because you have a neurodevelopmental disorder doesn't mean you stop developing. You continue developing, but maybe on a different trajectory. So if you're simply asking, is this child improving? Well, almost certainly yes, right? But what we're really trying to measure is if this level of improvement is different from the level of improvement on an active drug. And then unfortunately, like the uncertainty principle, what we ask can have an effect. And the clearest impact of this that I've observed is doing this really well-anchored scale, the Vineland Adaptive Behavior Scale, where you ask things like, listens to a story for at least 15 minutes. Almost all the time, or all the time, some of the time, or never. So you ask a question like that, and the parent says, I don't know, some of the time, and then they go home and they check. And then they come back and they now have a different answer because they tested it. And you see this over and over again. We've had families say, you know, we figured out he actually can tie his shoes, okay? But they've figured that out. That isn't an effective treatment. And then our care, right? And we are caring people even during a clinical trial, can have an impact. So have you tried this? He does really well when I do it this way. She seems like she's talking more this week. These sorts of statements, which we try to avoid, but are really difficult to keep away from completely, have an impact on how things go for a child. So as we try to move forward and test things that we hope have an impact, we have to mitigate this. So part of it is always having a placebo or an active control. Many of our behavioral studies use a waitlist control, which is actually telling somebody you're not going to change until. So that's sort of, you know, the opposite of a control, and really has been a challenge in ASD. You need truly blinded raters. So if you have dramatic adverse events, a recent study that got a lot of press this week of bumetanide, which is a diuretic that then results in people needing potassium supplements, it had very little placebo effect, dramatic effects of treatment, but of course almost everyone realizes they're on active treatment when their potassium needs to be supplemented. So real challenges there. Longer duration of treatment can mitigate some of the risk with simple regression to the mean, and then ideally equal randomization to treatment so that there isn't the expectancy that people are taking medicines. Outcome measures also need to be anchored as much as possible or observed instead of simply rated. That also helps us understand what's actually improving. And then we have to be careful to match our population and outcome measure. So in one of the studies I'll tell you about in a bit, it was difficult to measure cognition using the tasks that we were applying in a population with an average IQ in the 40s. So you have to find something that actually has a reasonable range in the population of interest. So one interest in dealing with this problem has been the idea of flipping the study design. This has been talked about for years, and then Forest Pharmaceuticals, and we were involved in this study, actually tried it. So they had everyone take active drug and then get randomized to either placebo or continuing on active medicine. And what they found, this has been presented but not published yet, was a strong nocebo effect. Even if you were continuing on active medicine, more than 60% of the time, you saw a significant worsening. So it can go in both directions, unfortunately. But we do have some history in the behavioral literature, particularly for people using applied behavior analytic sorts of techniques, of doing multiple baseline design, achieving one new baseline either without treatment or with treatment, seeing that somebody is stable, and then flipping treatment groups, the A-B sort of design. And you can see this done with a behavioral treatment here, without treatment, with treatment, without treatment, with treatment. It doesn't actually even matter what this axis is. But if we took this sort of approach, but randomized without the investigator or the family knowing, we might be able to avoid this problem, particularly in small treatment groups, which is what we're going to be stuck with in precision medicine trials. So let's pivot to talking about that, as I think maybe the most exciting pathway forward. And here are any number of things I could be talking about. I'm going to focus on one in the interest of time, which is Fragile X syndrome. So Fragile X syndrome doesn't always lead to autism spectrum disorder, but in somebody with Fragile X and autism, if you took the Fragile X away, they would almost certainly no longer have ASD. You do see mild to moderate intellectual disability in everyone with the diagnosis. You also see other behavioral features. I'm very briefly going to move through the molecular genetics, which has now been known for 20 plus years. There's an expanding trinucleotide repeat in the 5' untranslated region of the FMR1 gene. When that expands upon transmission to more than 200 repeats, you get hypermethylation of the gene and silencing, loss of the protein. I'm not going to focus directly on the protein's function, which is really as an RNA chaperone, regulating translation, as well as probably shuttling from the nucleus to the cytosol. But what I'm really going to focus on is a hypothesis that's emerged over the last 15 years from work largely by Mark Baer as well as others. So this role of FMRP is now reasonably well understood, that in the context of a glutamatergic synapse, whereas we oftentimes think about signaling at AMPA and NMDA receptors, FMRP actually lies downstream of mGluR5 signaling, such that if you have stimulation of mGluR5, FMRP actually shows increased translation and serves to dampen further signaling. Included among that signaling is the effect of removing AMPA receptors from the recirculating pool, such that if you don't have FMRP, you get too much removal of AMPA receptors, which presents as excessive long-term depression, resulting in synapses that fail to mature. And so in that context, you would expect to see excessive nearby dendritic spines and few dendritic spines that actually show a mature appearance. And that's exactly what's seen both in the brain of people with Fragile X syndrome as well as in the mouse model. And shown here, you see excessive dendritic spines that have this long filopodia-like appearance, whereas this is the typical brain shown here. These mice actually have a very mild behavioral phenotype with almost undetectable deficits in cognition, which is a real challenge when you're thinking about a model of a disorder that always includes intellectual disability. And their social deficits in some studies aren't present at all, and in other studies are very subtle as well. So the good news is that in mice, at least, in several studies, you can show that manipulating this pathway by blocking or tuning down the mGluR5 receptor, you're able to rescue multiple phenotypes. So this is work, probably the most comprehensive study done at Roche in collaboration with Mark Baer's group, where they gave an mGluR5 negative allosteric modulator starting shortly after weaning and extending all the way to sexual maturity. Obviously, that's hard to parallel in a human, but what you see in these mice is amazing. So restoration of the dendritic spines, shown here, where it's really indistinguishable from the wild type, improvement in hyperactivity, which is quite mild to begin with, improvement in some forms of learning, improvement in auditory sensitivity, decreases in seizures that can be induced in these animals, and importantly, no obvious ill effects on health. So tremendously exciting, the sort of thing that you'd like to then move into humans, and really the furthest that we've gotten along this sort of cascade, where you have effects at synapses. There's still some uncertainty about brain region, and then unfortunately there's some lack of clarity about how that actually translates to behavior, but some evidence, at least at the level of hyperactivity and learning tasks that don't easily translate across species. So based on that data, Roche and Novartis invested in large efforts to test similar drugs in humans with Fragile X syndrome. Now, because of the regulatory environment, you have to start in adults and then move down to adolescents and move down into older children and then younger children. And these are the results from Novartis for adults and adolescents, split in a couple of different ways. But you see, and I realize it's difficult to see, you're not missing much. In gray is the placebo response. And in one condition, placebo was actually significantly better than active drug. But in no condition was active drug better than placebo. And again, sliced in any number of different ways, really quite discouraging. However, very few of us would have said 10 years ago, you know, if you really want a medicine for a neurodevelopmental disorder, let's see if it works in adults. and let's just look at general measures of aberrant behavior, which was the easiest thing to look at in this study. Instead, you'd say, let's start in children, and let's try to capture a time period when you can see active learning and where we know something about how that looks, and perhaps even about how interventions can help spur learning. So I think there are a lot of questions about why these studies failed, but we're involved in a child study that we finally got the network-wide Notice of Grant Award. And that study is actually looking in young children, coupling medication together with a standardized level of language enrichment delivered via the parents, with the hope that this will be a way to actually parse the central question, whether this is actually something that moves learning in a way that can really be measured. So this is sort of like this picture where we know this from mice, and this study is looking early in development, taking that into account, and we're looking at behavior. We're also looking at information processing and using EEG to look at circuitry and processing at least of auditory stimuli. So it doesn't completely connect, but the idea is this will give us a clearer sense of whether this is a hypothesis that will carry us forward. I should say there are lots of other potential targets in Fragile X syndrome. It turns out that it's not true, but many things, not almost anything, but many things will at least rescue the dendritic spine phenotype in these mice, which maybe tells us that dendritic spines are not the first thing to look at before you're translating, that you want to see something else move. This cascade isn't really that different from the precision medicine cascade in cancer. To this point, right? So animal model, discovery of likely mechanism, reversal of that mechanism in the model, carried forward to clinical treatment studies. The difference is in cancer, you can look at whether the tumor is shrinking and whether somebody is surviving. We don't know yet what we're going to be able to anchor to. So we have a lot of questions there. And I think obviously with precision medicine, we hope to make an impact for individuals with the defined syndrome. But my hope in addition to that is that we're able to learn how to do these studies, right? So how do you actually translate something? And you could use Rett syndrome as an example of how to translate. You could use Fragile X syndrome. I don't know which one of these will give us the first clear lead on outcome measures that work, time points in development that work, how to develop this sort of study in humans. And then, of course, there's also the hope that you can translate beyond the syndrome, sort of thinking about the syndrome as the model for autism spectrum disorder. But I would say that's quite uncertain. So from there, sort of the opposite approach, the symptomatic approach. And this is really what we've got, right? And I could substitute in psychiatry. This is where we've had success. In ASD, the first thing I do when I see a kid is rule out medical conditions, and where they're present, treat those first. Then you can think about language, cognition. These are things we're able to see movement in with early behavioral intervention. We're not usually moving core symptoms of ASD, co-occurring psychiatric disorders, but even things like sociability. So whether a child prefers to be around another person or not, are not specific symptoms of autism spectrum disorder. Lots of kids with ASD like to be around other people. They just struggle in that interaction. So we can come up with ways to target these individual symptoms. But I would argue that that sort of approach, which is really behind the RDoC idea in a lot of ways, is likely to help maybe a larger group of children than an individually targeted precision medicine-like approach, but probably makes a smaller impact for each of those children taking the medicine. Certainly, these are not the sort of things that you would describe as cures or disease-modifying, but they are the sort of thing that you'd like to be able to offer, and this is all we have experience offering to date. That cascade maybe looks a little bit like this, where you have some information below behavior, but not a lot. Maybe you get all the way down to circuitry. At this point, not clearly. But we have some examples of treatments or potential treatments that are being carried forward that fit this sort of a model. So A lot of interest has focused on oxytocin. It clearly impacts social function across species. Some of that relates to sex and childbirth, but you can see in human studies that you can impact in some paradigms things like trust, social engagement, attention to social stimuli. There's also data looking at vasopressin, which you might think of sort of as a brother hormone, more effects in males than in females, versus oxytocin, better studied in females than in males. However, in autism spectrum disorder, there's actually very little evidence showing that there are deficits or somehow disruptions of the oxytocin system. So this is really more of the top-down thinking about circuitry, neurochemistry across species, things that may push on social behavior, may push on repetitive behavior, but aren't necessarily causal in the syndrome. There is some data in the laboratory setting showing that oxytocin delivered intranasally can make an impact for children with autism spectrum disorder. This is one such example. These individuals with autism spectrum disorder are either receiving intranasal oxytocin or are receiving placebo. And this is a typical comparison group just to show that the task works. And you can see that the individual can either throw the cyber ball, the pretend ball, to someone who throws it back, someone who throws it willy-nilly, or someone who never returns it. In the typical population, clearly a preference for playing catch with the person who plays catch nicely. In ASD receiving vehicle control, there's no such preference. And when they're receiving intranasal oxytocin, single dose, blind administration, you can see that there is a preference. So it can move behavior, not in every study. I'm picking one where you see things move. But that's exciting, right? And then there was this study which actually got more press, even though the behavioral component of the study was negative. So they were hoping to see a change in behavior. They didn't. What they saw instead was a change at the level of fMRI bold signal. And here briefly, they're again receiving either a single dose of intranasal oxytocin or placebo with a crossover design, so you can do within individual comparison. And then they're viewing either eyes or vehicles. So social stimulus, non-social stimulus, and then showing the differential between when they received the intranasal oxytocin versus placebo. And you can see a shift when viewing eyes in response to oxytocin versus placebo in some areas that are really interesting, like the nucleus accumbens, as well as a number of areas they liken to the social brain, but also in some areas that are perhaps a little harder to tie together. But this shows that you can see a difference in brain response in response to intranasal oxytocin. So you've got brain behavior, you don't necessarily have them in the same individuals, but clearly it can do something, including doing something in people with autism spectrum disorder. Some of you have seen this slide before. I use it as maybe the parallel to the secretin story. So you can now receive intranasal oxytocin via Amazon Prime in New York, probably within 24 hours. This is not what we should be doing. Just because it does something with single dose administration in the lab doesn't mean this is something we would describe as a treatment. Cyberball deficits are not a symptom of autism spectrum disorder. For those who are curious, it also may be useful in getting people to trust you or restoring interest in a mate. Not really. But we need to move this now to the real world setting. We've had lots of single dose administration studies, some short-term studies that by and large are underpowered. And so this is where it would maybe be nicer to be giving this talk next year versus this year. I could tell you the outcome, but we will have our randomized controlled study complete in April, and then we'll do data cleaning, and hopefully by the summer have initial waves of analyses, multi-site study, a number of groups. It's been really fun to be involved in this study, but because of the concern about placebo, we spend a lot of time reminding families over and over again, you could be on active medicine, you could be on placebo, and now say that every time the family says something positive, we respond, you know, we still don't know if you're on active medicine or placebo, to try to buffer some of that dramatic response to placebo. So it's actually almost 6 months. So it's a 24-week study, randomized portion, and then we actually have an open label extension that's another 24 weeks. And part of the idea behind the extension is some preliminary data from Lynn Sikich's group showing that they did a shorter duration and then had an open label extension. They saw separation between the placebo-controlled groups, actually once they were already in the open label extension phase, suggesting that you may need a prolonged duration of treatment, but that's preliminary data, and I think it's important not to draw too many conclusions from that. So that takes me in 10 minutes. So I'm not probably going to get to my restricted interest unless somebody wants to ask me a question about the serotonin system in autism. Hint, hint, I should have bribed somebody in advance. But in any case, I want to think with you a little bit about trying to bridge the gap between these symptomatic sorts of treatments and the precision medicine approach, because we really have a challenge in trying to evaluate treatments that may benefit more than the 1%, but also refine the folks who may benefit from a particular neurobiology-based treatment. And I'm going to use an example of this that was just published, although I've talked about it for some time. I'm not going to focus on the mechanism. I'm not going to say this is something I think works. I'm going to show the data as real data because I think it's an example of the challenge that we're likely to run into over and over again. So this was a study where the primary outcome measure did not show a difference between the active treated group, taking arbaclofen or XTX209, and the placebo treated group. There was movement in one scale, and I'm going to zoom in on that in a minute. But to just convince you, there is no difference here between the active treated group and placebo on parent ratings of social withdrawal and lethargy. And really that scale was chosen because it has good psychometric properties, not necessarily because it's a great measure. But in addition to that, there were some general scales, clinical global impression of improvement and clinical global impression of severity that were secondary outcome measures. And the severity scale actually showed a significant difference at the end of treatment. And this is change seen versus baseline. And you see here that there's separation already by about 8 weeks, and it was only a 12-week trial. When you zoom in on that and really think about who's driving it, the most apparent difference is the kids who don't have a change in this 7-point scale of severity, where you see there are more kids on placebo versus kids on active treatment who don't show a change. In terms of kids who do show a change, where you see some difference is at the level of big shifts. These are the sorts of shifts we don't commonly see in the clinic, but these are the sorts of shifts we'd love to see, but it's a tiny little component of the sample. And truth be told, if autism is as heterogeneous as I told you, and you do a study of all of autism and have a treatment that's actually based on an understanding of neurobiology, probably just in a subset, this is the sort of thing that we would expect to run into over and over again. So, yeah.
Is that the same compound
So is this the same compound as Jim McCracken's group and the FAST AS group we're studying? No, so they were studying a GABA A agonist that has some selectivity, and they had essentially a charge to show target engagement, which they showed, but they didn't really have power or a charge to go after specific treatment effects, so effects on behavioral symptoms. And so they had a pretty general ASD population and didn't really see a movement in symptoms. And my understanding is that that's unlikely to be funded going forward, which is a real challenge as we think about these fast fail designs. We have to keep our eye on target engagement, if that's what you're trying to do, and let go of the idea that we're moving symptoms in the initial study. Or if we also have to move symptoms, we have to enroll people who are more severe in the symptom domain that's of interest to the study. And I think it's an unfortunate lesson, but I think it's an important one. That's a great question. So their measure of target engagement was actually an EEG measure, where they had a few different EEG measures. And Jim hasn't presented the final version of that, so I don't know that I can tell you with a lot of clarity about what EEG parameter they ended up picking. I saw some of the early stuff and heard some of it later. So he's presented some of the initial data. So obviously, I'm not saying that this is a treatment to initiate in kids with ASD, but I think it really illustrates our core challenge as we're thinking about ASD, which is that we need to figure out who's likely to respond to a treatment. And ideally, that would be at the level of neurobiology or some marker that's anchored in observation that connects to our medication mechanism of action. So in this study, there actually was a clinical profile of kids who tended to be in that responder group. They tended to have higher IQ, they tended to have better language. That may actually tell us something about our outcome measures and the ability of our outcome measures to detect change in particular subgroups of the ASD population. But what we'd really like is something at the level of like what the McCracken group was looking at, an EEG or an MEG marker that you can see changes in response to treatment or shows a baseline difference. And you'd like that to be something that obviously you can measure reliably. But it's really quite unclear whether it's something that needs to be tied to genetics, whether it's something that needs to have a clear demarcation between people who have that feature versus people who don't, whether it needs to change with treatment, whether it really needs to connect to pathophysiology. Really, it's something that needs to give us a clue about treatment. And so we need to start thinking about biomarkers as we're going into treatment studies where we think we do tie to neurobiology, but where we don't necessarily know what subgroup is going to move. If this is sort of our model of current treatments, where we have symptomatic treatments who have a larger percentage of people who are targeted or can be targeted, but a smaller impact of treatment, and precision medicine, which right now is hypothetical, but could have a huge impact, but in a very narrow slice. We'd hope that biomarkers fit somewhere here in the middle. Exactly where is hard to say and probably differs based on biomarker. We have a number of, I would say, quite reasonable clues about biomarkers. So on the genetic front, there's consistent evidence that there are more genes bound by FMRP, the Fragile X protein, that are associated with autism spectrum disorder than you'd expect by chance, suggesting this may be a hub from the perspective of autism or neurodevelopment. There are another couple of genes that fit that same sort of pattern. Unfortunately, the directionality remains a concern. So some of those genes actually look like they're downregulated at the translational level by FMRP, some upregulated, so you may make some people worse while you're making others better. There are interesting neuroimaging biomarkers. I would say I myself am more hopeful about things that we can actually perform in kids with ASD versus MRI studies, which are really challenging and give us a pretty narrow slice of autism spectrum disorder. Kids just have a hard time lying still. There are some interesting whole body symptoms. So generalized overgrowth seems to be present in a subset of kids with ASD. There are some immune markers. And then increasingly, we're understanding maternal risk factors related to autism spectrum disorder. So one of those is maternal antibodies present in about 20% of kids with ASD. So, and I'm actually going to, oh gosh, this is the hazard of doing this not on my laptop. But I'm going to move forward, acknowledging that I don't have the 10 minutes to give a full discussion of one example biomarker, although I'd be happy to come back to that. I have 30 minutes left? Okay, well, I'll go back and go. So I'll go fairly quickly through this. And I really intend this as an example of the sort of thing that you can do with a biomarker or where it may give you clues. So this biomarker is better characterized than most biomarkers that are described in ASD. That doesn't make it the best, but it's certainly been around the longest. And what you see here in white is the typical bell curve of serotonin levels that you'd expect to see in the general population. And in yellow is the second peak in kids with autism spectrum disorder. This is the most homogeneous sample that has been reported to date. Oftentimes it just looks like a big tail. But certainly there is this group in ASD that has a marked elevation in blood serotonin levels. those serotonin levels are contained within platelets. They're not floating free in the blood. Serotonin gets into the platelet via the serotonin transporter. Serotonin levels are more heritable than autism spectrum disorder itself. And so we've done some work mapping genes that may underlie serotonin levels as a biomarker independent of autism spectrum disorder. But interestingly, back in the days when people did linkage studies in autism spectrum disorder, the strongest linkage peak ever described in autism spectrum disorder actually overlies the serotonin transporter gene, described here as SERT or SLC6A4. And that linkage peak is driven exclusively by families with 2 or more affected boys. And no affected girls. If there's 1 or more affected girls in the family, you don't see any signal whatsoever. And interestingly, when we mapped the biomarker, we found that the serotonin transporter gene was associated with serotonin levels only in males, not in females. So underlying that linkage peak were a number of rare variants of the serotonin transporter. Each of these leads to an increase in serotonin transport in cell models. And that would be consistent with sucking more serotonin up into the platelet. And so that seemed like a reasonable lead. Only one of these variants was common enough to even look at it by itself, and that was this glycine to alanine change. It's in a regulatory region of the protein. You can see it's conserved as glycine in mammals, but it's actually alanine in birds. Within these families, there was association with rigid compulsive symptoms and sensory aversion. Many of these families, though, had this sort of a pattern, where females in the family carrying the variant did not have autism spectrum disorder. They were not necessarily ascertained for other psychiatric disorders. And then you see multiple affected males who carry the variant. This is a complicated gene and protein. There's a lot of work that's been done on it. The advantage of this variant versus a lot of the common variants that have been studied extensively is that it actually can be moved into mice in a much more straightforward way than something that regulates the level of transcription. And so that's exactly what we did. And this recapitulated the biomarker where you see elevated blood serotonin levels in the mutants compared to wild type animals. This is published data. I'm not going to spend any time on it. But we then looked in male animals only and did see abnormalities in social behavior, did see abnormalities in repetitive behavior. I wouldn't necessarily describe this as autism-like, but it at least would point you to impacting the systems that you think might matter in autism. And then we turned, because of that sensory aversion finding, and what I'm about to tell you about, to initial studies looking at development of the sensory system. So the serotonin transporter is actually transiently expressed in a number of sensory regions, even though in adult animals it's really only expressed in the RFA. So we focused on somatosensory development because first, it's easier to do, and second, usually sensory aversion in autism is in the tactile domain. And so we had the hypothesis that some abnormality happening over the course of development was somehow disrupting the somatosensory system, leading to sensory aversion. And we actually looked quite early after some time spent looking anatomically to understand whether there were differences in the whisker barrels, the easiest way to read out the somatosensory system in mice, and then looked at this early time point when the serotonin transporter is expressed and where data shows that serotonin actually modulates response to other axon guidance cues during the period where projections are coming from the thalamus into the cortex, and particularly seem responsive to serotonin on the somatosensory side. And then sort of happy, although at the time I thought it was an unhappy accident, we had difficulty getting a large enough sample size for pilot work in animals bred the proper way, which is looking at heterozygous dams that generate either a wild type embryo or a mutant embryo. And this controls for the DAMS genotype and ideally avoids that genotype impacting what you're finding. But then my graduate student was having difficulty getting enough embryos that way, so he did a few of these breedings where you're using a wild type dam to generate a larger set of wild type embryos, or a mutant dam, a larger set of mutant embryos. And what we saw, we were initially excited about, which is that we seemed to have a wider breadth of these trajectories, suggesting that there did seem to be an impact of genotype. But then as we went back through it, that seemed really quite inconsistent from animal to animal, and we saw a lot of variability. and finally figured out that we needed to separate by breeding scheme, and that by doing that and then repeating in independent cohorts of animals, we actually were able to pull apart a maternal genotype effect that was separate from an embryo genotype effect. So you don't see any difference based on embryo genotype at this point in development, whereas you do see a difference driven by dam genotype. So the embryo genotypes obviously are still opposite wild type and mutant, but being housed in dams of different genotypes. So that wasn't exactly what we were looking for, and was particularly a puzzle because the serotonin system during embryonic development actually has multiple sources. Early in development, you actually see that the placenta is responsible for serotonin in the forebrain. And so shown in blue is serotonin in the forebrain derived from the placenta, and in red are the projections coming from the RFA, finally making it into the cortex, certainly responsible for the vast majority of serotonin in the cortex by the time of birth. And so we looked at that time point in development where the placenta ought to be responsible for serotonin levels. And in the placenta, we saw that our mutant dams had embryos with lower levels of serotonin in the placenta. And in the forebrain of those embryos, you saw lower levels of serotonin, consistent with the idea that the serotonin is coming from the placenta. And in the hindbrain, we saw no such difference, again, consistent with the idea that there are different sources. When we looked at mutant versus wild type embryos housed in heterozygous dams, we saw no difference, showing that it really doesn't seem to be driven by the embryo genotype, which is the same genotype of the placenta, which is really quite bothersome. So when we carry this forward to understand or to solidify the relationship with placenta, looking at a time point in development where now those projections have reached the cortex. Now in the forebrain, we don't see a difference, even though we continue to see a difference at the level of placenta. So there's a lot more work that we're doing to try to understand that mechanism, but I just want to pivot to sort of the way I think about why we do work in mice, and something that's always important to know, which is this actually relevant in the human, or does this turn out to be just a really neat, quirky finding that we turned up in mice by accident? So ideally, you do that at a genetic level, but this particular variant is rare, and then there are many other variants of the serotonin transporter gene that affect expression, but in a complex way, meaning you'd have to take multiple variants into account. On the other hand, this biomarker may be a reasonable surrogate to think about the impact of the serotonin system from the maternal side. And so we developed the hypothesis that maternal whole blood serotonin levels, not the way we usually think about this biomarker in the kids, would be associated with the phenotype in kids. Unfortunately, we didn't have access to blood during pregnancy in a sample that actually had longitudinal diagnosis of autism, sort of a heavy ask. And we didn't have access to maternal serotonin levels in a general population sample. But what we did have access to was serotonin levels in every member of families where the child already had an ASD diagnosis, and this is in collaboration with Ed Cook's group at the University of Illinois. And in this sort of setup, it's important to recall that serotonin levels are heritable. So there is a correlation between the child's serotonin levels and the parent's serotonin levels, both for mom and dad, a little more with dad. And it's also important to recall that every kid has autism spectrum disorder. We don't have a controlled population here. So we must always think about the effects of the child's biomarker, and then we also have to think about the effect of dad separately from mom, but sort of have a built-in control, where dad has the same degree of relatedness to child and usually shares ethnicity, although not always. And then we wanted to use well-anchored measures, autism diagnostic observation schedule, direct observation of the child as a social or repetitive behavior measure, vocabulary measures, adaptive behavior scales, and then started looking in a really exploratory way. The first thing is to understand whether the biomarker in the child relates to phenotype, and it doesn't. And actually that's been described over and over again, really quite bothersome. And then we started looking at the parents and thinking about dad as the control for mom, saw a negative correlation between a social observation on the autism diagnostic observation schedule, negative correlation for repetitive behavior, positive correlation for 2 different measures of both receptive and expressive vocabulary, changes in adaptive behavior. So it's getting spooky at this point. Nothing showing up in dad, only seeing correlations in mom. Now, importantly, we didn't use mom's ratings of the child because we were concerned that that could have an impact. And we don't have association between serotonin levels in the parent and their behavioral measures either. So these are not deterministic sort of correlations, but they are statistically robust. And here too, we have lots left to do. So on the mouse level, we're really going after a mechanism to understand why this happens. We know it isn't simply at the level of the rate limiting step in serotonin synthesis in the placenta, but doing a lot more work there. And in humans, because it is this broad exploratory set of findings, we're starting to do latent class analysis to try to see if this boils down to something that can be understood more clearly, and then clearly it needs to be replicated. Lots of questions about where to go from there. And let me briefly talk about sort of the last of the many challenges, which is ultimately we're not going to have drugs that just do something by themselves. We're going to have to put medication or other biology-based treatments together with behavioral treatments. This is really the only significant example of that to date in autism spectrum disorder, and that is a comparison of 2 groups, one of which is taking risperidone plus also getting behavioral treatment, and the other group of which is only getting risperidone alone. You can see both groups show dramatic improvement in disruptive behavior, but there is a significant advantage for the behavioral treatment. Unfortunately, that advantage goes away 6 months later, so it doesn't appear to be sustained. But this maybe tells you something about the challenges of combining 2 things when one of them has a huge effect. You probably need to use a sub-threshold level of medication treatment plus behavior in order to pull out effects of the behavioral treatment alone. What we'd prefer, and this is sort of still a controversial area, is something that potentiates treatment response a little more like how some of us think about D-cycloserine with exposure-based psychotherapies, where you have a treatment that potentiates response. And then usually in those studies, you see the biggest difference at mid-treatment. So sub-threshold level of behavioral treatment plus medication actually allows you to see a medication effect. And I suspect we're going to have to develop a whole methodology around how to put these things together for combining things like oxytocin plus social skills training, assuming oxytocin does something. So thank you for the indulgence of the extra 10 minutes. This is a summary which I won't read off. I would say we do have challenges. I would say we also have reason to hope. I think we will see things carry forward in precision medicine. And I think we will learn about how we do these sorts of studies more effectively. Lots of people to thank. I'm not going to be able to call out by name, but a lot of folks bridging my time at Vanderbilt and then here, and then a lot of folks whose work I didn't do justice to but glossed over quite quickly on the serotonin side, and then finally sources of funding. So thank you very much for your attention.
Turn these on. Jeremy, thanks for that really extensive description of your view and work to date in autism. I tell you, autism research is not for the faint of heart.
Nor is schizophrenia research. We work in challenging areas.
Yes, yeah, but I think autism is really the ultimate black box. So, sort of my first question is that Dustin Hoffman and Rain Man did for autism what whoever the actor was that played the Elephant Man in that play for neurofibromatosis, because it wasn't a known entity, and so autism was first described by Kanner when in the 1940s.
1943.
And you showed Fragile X was first described in 1930.
In 1943 as well.
So there are these genetic developmental disorders have sort of been hiding in plain sight. What were they called before? I mean, how were those people thought of?
Yeah, so that's a great question. So first, a note about Rain Man. So that movie was based on Kim Peek, who has been sort of described in the popular media, so I'm not disclosing something, but Kim Peek actually has a genetic disorder, FG syndrome, that has a defined set of features and sometimes includes autism spectrum disorder, and interestingly, sometimes includes psychosis as well. And I think a lot of the folks with these rare things have been hiding within our description of other things. So you often find in these studies that are in schizophrenia or in autism spectrum disorder, the same sorts of variants pulled out. And then if you look back, you find that the individual, say, in the schizophrenia group who has a 16p11 deletion, turns out to have had neurodevelopmental delay and social difficulties, and has a lower IQ than their family members, and now has psychosis. And I think that is by and large the way we should expect biology to work, that we will describe what somebody is experiencing at any point in developmental time. Velocardiofacial syndrome is a good example to talk about here, is another such example. So we see kids with velocardiofacial syndrome that have developmental delay, ADHD, and sometimes autism spectrum disorder. And then in adolescence, frequently they present with mood symptoms. And then obviously in adulthood, you'll see them in a schizophrenia clinic. So I think some of those genetic disorders are hiding within our descriptions of other things. I think a bigger question is, where has autism spectrum disorder been in human history? And there are descriptions that are quite elegant, going certainly back into the 1800s, and some even further than that, where if you read it and think about the sort of role of society at that time, they were probably elegant descriptions of autism spectrum disorder based on concepts at that time. But it's not that easy to find. You find in societies that don't do a lot of diagnosis that many people with neurodevelopmental disorders are cared for within the family and community context without necessarily getting a lot of schooling, without getting a lot of treatment. They tend to do better in some ways, meaning they tend to be less distressed, in part because the family figures out what they can and cannot do and has them do that. Whereas expecting kids to go through schooling can sometimes lead to more distress. So I think that's an attempt to answer. I think the flip side of your question is about, which we get asked about a lot, the increase in autism diagnosis over time, that I would say is also hard to evaluate because you can't go back in time, but is largely driven by changes in ascertainment and diagnosis, how we diagnose, how we screen, who we characterize as affected, as opposed to something that really is a societal increase in a disease. There may be real factors underlying some increase. So extreme preterm birth with very low birth weight. We see a lot more babies survive that now. That's a good thing, but that dramatically increases the risk of autism and a bunch of other neurodevelopmental consequences. And there are some other risk factors that sort of fit in that category.
Yeah, including one who's our president, paternal age, right?
Yes. That, yeah. Although, yeah, the other part of that I won't comment on.
Don't say any more about that.
Yeah, but yes. So there is abundant evidence that paternal age is a risk factor for neurodevelopmental disorders, for autism spectrum disorder. A little question about schizophrenia. I mean, not just a question, a little evidence for schizophrenia. And it isn't just that. It also is probably grandparental age. So there's some evidence that your mother's father's age at her birth may be a risk factor for autism spectrum disorder and probably neurodevelopmental disorders. Why would that be? Well, there seems to be
in the grandchildren.
In the grandchildren. So grandpa, mother, child. It isn't a ton of evidence. It's sort of at the level of what we're seeing now for schizophrenia, but it sort of makes sense. Being female is protective against autism spectrum disorder. Actually, it's better for your social skills generally, at least mine. But it's not surprising that you would see things that wouldn't lead to a phenotype in women, but then might lead to a phenotype in men. And there may be flip sides to that, but this is something that's getting a lot of attention now.
Well, is that to say that grandparental age would be penetrant in male offspring who are parents but not necessarily as much the female?
So that's another issue. So we don't really know what that parental age risk factor is about. Some of it is clearly about mutations. Some of it is probably about epigenetics, and I don't know if Jay is here, but Jay Jay Gingrich has done some really interesting work showing that there are epigenetic differences in sperm based on age of at least the mouse parent, mouse father, and that is one of the ideas behind this. You also see imprinting effects where there may be a particular genetic mutation that only is going to manifest if inherited from mom or dad. And there are examples of that within autism as well.
The sociologist at Columbia who has done work in this area, and he's given grand rounds, isn't it Pete or something?
Peter Bierman.
So, I mean, his contention would be more along the lines of what you're saying. It's social factors lead to increase awareness, increase identification, and broadening of the boundaries.
Yeah, so there are all sorts of interesting hypotheses about that effect, including the idea that men who reproduce later may have had more difficulty finding a mate. And so it may be that indicates something about traits related to social function that may relate to risk. I wouldn't say that we have evidence that that's the case, because you see those traits running in families regardless of the age of the father when they had a child with autism. So it's a little hard to separate that. But there are all sorts of factors that could underlie that, some of which are still quite controversial.
Just coming back to the comments before about conditions embedded within, syndromes or broader conditions. To anybody who has to respond to critics about the proliferation of psychiatric diagnoses, like DSM has 265 now, remind them that the first effort to try and quantify the population frequency of mental disorders in 1840s, the two categories on the census were insanity and idiocy. So I think in that sense, the kind of peeling of the onion and more precise definition has been, an unquestionable sign of progress.
Yeah.
And more to come.
Yeah.
Okay. Well, I don't want to monopolize this. Who else has comments, question? Yes.
One of the issues that would be is interesting in this field is the ratio, the gender ratio, which would have biological implications. I also had another question, and that is that, did you say what is known about Geodon in terms of clinical trials for autism symptoms?
Sure.
And then the fact that maybe combining some of them might have better effects and might also have implications on what is underlying.
Okay.
The neurobiological underlying.
Second question first. We essentially know nothing. There are no placebo controls of ziprasidone in autism spectrum disorder. You can believe that the overall class of medicines may show benefit, but there is one negative trial of lurasidone in autism spectrum disorder, and it really is negative. So it clearly is not just a class effect, but certainly people have used ziprasidone. The other question, so I didn't really focus on the overall epidemiology of autism, but autism, as I think we all know, is more common in boys than in girls, dramatically more common. And actually the ratio gets bigger the higher the IQ. So that if you find kids, and the data aren't perfect, who have an IQ of like 145, there's something like a 10-to-1 ratio. Again, data aren't perfect. If you go down to an IQ in the 40s or so, the ratio approaches 1-to-1. And so that could suggest a number of things, but certainly suggests that there is a protective effect of being female, at least of autism happening alone, without intellectual disability or significant cognitive impact. And I think there's a lot of interest in that, and there actually now is a study being sponsored by the Autism Science Foundation looking at sisters of boys with autism or mothers of boys with autism who also have a sibling with autism. Alex.
Thanks, Jeremy. Do we know how much de novo mutations contribute to the variance?
So increasingly, and I had one slide dedicated to popping up some of the emerging data on de novo variants. It's actually really hard to pull out the odds ratios for the de novo variants because they are so rare. So the degree of risk attributed to any one of those de novo variants remains quite unknown for the majority of them. Based on how you approach the overall group of de novo variants, you would say that de novo variants account for, and it depends on who's drawing it, somewhere between 5% and 20% of individuals with autism spectrum disorder, which is to say you can find them in that group. What's less clear is the degree of risk that you can attribute to them in any of those individuals. Because of course you do see some de novo variants in the general population, and you're really based on your overall ratio of de novo variants in ASD versus by and large unaffected siblings, and doing that at the level of all of the de novo variants as opposed to variants in individual genes. There are exceptions to that, but at this point we're still at the level of finding something 3 times as a de novo variant and saying that's an autism gene. And it's an autism risk gene, but you don't know that if you took that away, the child wouldn't have autism. And in some of the studies that have looked across families, you'll find a de novo variant in one kid that's affected that's not present in the other kid that's affected. And well, what does that mean, right? So there's clearly something else that contributes to risk. And I think increasingly we need to start thinking about rare variants as susceptibility variants, as opposed to how we like to talk about mutations, which is A causes B. And it clearly isn't that sort of relationship, except for in the syndromal cases where really you're not diagnosing autism on the basis of that. You're saying MECP2 mutations lead to Rett syndrome, FMR1 repeat expansion leads to Fragile X syndrome, not to autism necessarily.
And I'm just, Jeff had to leave to take a call, so I have been drafted in. Please.
That was a fascinating presentation, and I'm thinking that in my series of 2, I'm a general psychiatrist. One kid has complete language, I mean bilingual, and very poorly related, and another kid has no language at all and is very well related. And so your emphasis... Very well related, interpersonal.
Okay, yeah.
Very attached, all that vasopressin, I guess it's a boy. You emphasize in a helpful and constructive way the tremendous complexity and the heterogeneity of what we all recognize as autism. My question is more concrete, actually, and that is how much of this is being taught? I was struck by Jeff's comment that the ultimate black box is not even schizophrenia, which obviously he knows a lot about, but autism. And I just wonder how much of this is being taught to residents, people who may be able to pick up on this. It surely belongs within the realm of psychiatry or psychiatry neurology. And I don't know how much people are really exposed to be able to relate some of this to what they're learning in the ordinary course of residency.
So we should ask a resident. That's not fair.
You can do that at lunch. I hope you will. Yeah.
Yeah. So I think this is something that's taught in medical school. I lecture on this to the medical students, and in that context, we include videos so that they get a sense of what autism looks like. I would say that the younger somebody is in their medical training, actually the more conscious they tend to be about autism spectrum disorder, in part because of media and early exposure. It is something that still gets missed. So frequently you see somebody present not having had a lot of healthcare access with what seems like psychosis, but really maybe agitation in the context of minimal verbal status, meaning they can't really express themselves, they're getting agitated. Sometimes I used to see those folks diagnosed with schizophrenia or a psychotic disorder, when the truth was that they were reliving an experience intensely that was maybe traumatic and clearly had autism spectrum disorder. And that can look a lot like psychosis. So I think we do need to be conscious of this. And in psychiatry, we need to be particularly conscious because particularly in folks who are over 20, but even over 30 currently. There are a lot of people who have autism spectrum disorder by DSM criteria, but were never diagnosed. So I do consultations with adults at the Center for Autism and the Developing Brain, and I've seen 40-something-year-olds who are presenting with decompensation after a loss in the family, have never had an ASD diagnosis, clearly have ASD, but have been able to do all right within a nice environment that their family has set up for them, and then things fall apart, and now they're really impaired at a systems or family level, whereas before that wasn't as obvious.
So I know there are probably many more questions, and there's lunch afterward. I just wanted to join the comments Jeff made at the beginning, which is obviously Moira made a fabulous pick. I don't know if she's still here, in recruiting you, Jeremy, and I think it's really the tour de force from the basic science through the clinical trials, and also what comes across really clearly is your clinical expertise, and that's a very rare package, I think, not just for a child psychiatrist, for an adult psychiatrist, but it is actually who I think our department is and can be, and so it's a really great fit, and it's a real pleasure to hear your talk.
Well,
so
thank
people
you.
who want to ask more questions, please go up to lunch afterwards.